everyone, and thank you for joining this special HPTN 083 Preliminary Studies Results webinar. HPTN 083 is a global randomized, controlled, double-blinded study that compared the safety and efficacy of long-acting injectable cabotegravir, or CAB-LA, to daily oral Truvada for pre-exposure prophylaxis. The study showed that CAB-LA lowered HIV incidence among cisgender men and transgender women who have sex with men. HPTN 084 is the first study to compare the safety and efficacy of CAB-LA to daily oral Truvada for HIV PrEP among cisgender women. A discussion on its importance to HIV prevention for cisgender women will follow. Some quick reminders for today's webinar. Participants are asked to not share their video as it slows down the webinar and causes challenges to those with lower internet speeds and bandwidths. The Zoom webinar is being recorded, and that recording will be made available for review at a later time. And again, all lines have been muted. Questions should be submitted using the Zoom chat feature. So now we'll introduce our moderator for today's webinar. For over 16 years, Mitchell Warren has been the executive director of AVAC, which focuses on HIV prevention, research advocacy, communications, and stakeholder engagement. He was previously the senior director for vaccine preparedness at the International AIDS Vaccine Initiative, and also spent 4 years as vice president and director of international affairs for the Female Health Company, the manufacturer of the female condom, where he directed efforts to design and implement reproductive health programs that integrate the female condom, and he led global advocacy efforts for expanded commitment to female-initiated prevention methods. Mitchell has also
Cheryl, that's, you don't have to go on. That's more than enough.
Thank you, Mitchell. You now have the floor.
I hated to interrupt, but you all joined a call to hear what I think are really exciting and important information, and I will also be brief with introductions to the 4 people you're going to hear from over the next 30 minutes or so. And most of all, we want to get the great questions and comments from one and all. So again, just to reiterate what Cheryl said, we are really keen to get your questions in the chat feature. There's also the Q&A box. I'll be paying attention to both. We're going to do our best in 1 hour to get to as many questions as we can. And I know I can't officially speak on behalf of the HPTN, but I think I probably can in this regard, that we are all at the PTN and at AVAC separately committed to making sure these questions get answered on an ongoing basis in the days and weeks and months to come, because this is a critically important conversation for the whole community. I also just want to say at the very outset how grateful I am to the HPTN for inviting me to join you all today and participate. And just to say, good news is always welcome in HIV. We don't get good news often enough, but I think everyone would say that in the last 5 months, good news has never been more needed in any form or fashion. So this week's flurry of information about HPTN 083 is especially welcome. I'm going to, just for the run of show, I'm going to, in just a moment, turn it over to the co-principal investigators of the HPTN 083 study to present preliminary results, which you've heard publicly, but to hear it from Rafi and Beatrice is just so important. We'll then, and again, please feel free to put questions in as they present. We're then going to turn to the co-PIs of the sibling study, HPTN 084, to get an update from them about that trial, which is more important than ever. And then we'll go through as many questions as we can in the course of the call. And again, I'm not going to get into super long introductions because what they have to say is, I think, far more important than who anybody is. But Rafi is the protocol chair of 083, as I think many of you know, based in California at the University of California, Los Angeles, UCLA, where he directs the UCLA Care Center and has been a leader in research and in clinical care. And I know he's been doing triple duty in the midst of COVID, and just delighted, Rafi, to have you on, along with Beatrice Grinstein from Brazil, from Rio de Janeiro, one of the nicest cities in the world. Beatrice has also been a long-standing clinician and researcher based at Fiocruz in Brazil, co-chairs the HPTN 083 study, and has been involved in a large number of both clinical trials, as well as has been a pioneer in oral PrEP programs in Brazil and in the world. So I'm delighted to first turn it over to Rafi and Beatrice, and then I'll introduce the 084 leadership, and we'll go to them. So Rafi and Beatrice.
Thanks, Mitchell, and thank you everyone for joining this morning. I know everyone has a lot of competing priorities, but we're really excited to have a chance to begin this conversation about the preliminary results of HPTN 083. And I do want to remind everyone that these are preliminary results. I'll be telling you a little bit more about the circumstances under which we were made aware of these results. This was an early stopping point for the study, and we did not necessarily expect to see results at this early time point. So there are a lot of questions that you may have, and certainly that we have, that we don't yet have the answers to. So we're going to be really grateful to everyone for appreciating that there are a tremendous number of critically important questions to the full understanding of these results that we don't have yet. So as exciting as these results are, there are going to be some things about it that are going to leave many of us sort of unsatisfied at this point. So we apologize in advance for that. So let's move to the next slide, please. So this is going to be our agenda for the 083 portion of the study. I'm going to briefly review the HPTN and its structure for those who don't know. And then right after that, I'm going to let my brilliant and wonderful co-chair, Dr. Grinstein, make some overall comments about what has been really accomplished thanks to the incredible partnership with community in the design and implementation of the study. And then we'll go into the preliminary results. Next slide. So again, an overview of the HPTN. I know many of you are very familiar with the HPTN, but just for anyone on the call who is not. Next slide. The HPTN is one of the NIH Division of AIDS funded clinical trials networks that's focused on non-vaccine HIV prevention. Sites are global, all over the world. You can see on this slide the regions and countries where the HPTN has ongoing activity, including the Americas, Sub-Saharan and East Africa, newly in Eastern Europe, and in Southeast Asia. Next slide, please. So just before I go into the study design, I want to ask Dr. Grinstein to have a chance to speak and to really contextualize what we're going to be talking about today in a way that really, she has an incredible facility and of describing it in a way that I think will be really important for everyone to hear. Beatrice?
Yes, so I just wanted to take a moment before Rafi starts with the preliminary results to thank, first to say good morning, good afternoon, or good evening, depending on where you are, and to thank first to AVAC and the HPTN Community Working Group friends for the organization of this webinar. We are all very happy and proud with the preliminary results of HPTN 083 and with this opportunity to present them to the broader community here in this webinar today. So these results for sure would never have been achieved without the profound engagement of the community. So I wanted to take this moment to thank and congratulate each of you for the enormous commitment and invaluable work you did in HPTN 083. So I wanted you to have this feeling with you before Rafi goes ahead and present the actual preliminary results. I am certain that the tremendous efforts you all did in the multiple participating sites in the US, Latin America, Asia, and in South Africa, when you reached out to the most vulnerable individuals and working so hard to retain them, has made all this difference. Of course, also the support and the engagement community had in the design of the study as well. Long-acting CAB as PrEP can be a great new tool for prevention that does not require adherence to an oral daily pill. And you know so well the importance of having options for everyone, taking into consideration their personal and life challenges. So I just wanted to pass on to Rafi so that he can provide you with the actual details of the preliminary results we have. And again, thank you very much for all your commitment and engagement with this, with 083. Thank you.
Thank you, Beatrice. And again, you know, Beatrice said it so beautifully. There's no way this study could have been conducted with this much relevance without everybody's collaboration, cooperation, and belief that enrollment of a population of most vulnerable populations and trust building with most vulnerable populations was possible and important and in fact critical to this work. So let's move on to the study design. Next slide. I think most people are probably familiar with this design. 083 was conceived of as a phase 2b3 trial, meaning it was still collecting safety data as well as designed to demonstrate efficacy of the investigational product, cabotegravir. Its design was that it compared once daily oral PrEP with Truvada to a regimen that included injectable PrEP with cabotegravir. It was randomized and it involved placebos, but no participant received only placebo product. That's really important. So it was what's called a double dummy study that was done in a double blind fashion, which means everyone received one active product and one placebo product during the blinded phase of the study, which is really critical to understanding the study design, because that reduces in the most rigorous way the amount of bias that the investigators could have and gives the best data to support regulatory submissions of a new product, whether a prevention or a treatment product. Next slide. This is a graphic of the study design. I apologize that it's a little bit complicated. Hopefully most people are familiar with this study design, but people were randomized in equal fashion, so in a 1-to-1 ratio to either group A, which is the cabotegravir group in orange at the top of your slide, or group B, which is the Truvada arm on the blue bottom of this slide. If people were randomized to group A, in the first 5 weeks of the study, they took an active cabotegravir tablet and a placebo Truvada tablet every day. So everyone took 2 tablets a day for the first 5 weeks. People who had good tolerability and safety, after taking the oral preparation in arm A, they continued on to step 2, where they received an active cabotegravir injection and continued the Truvada placebos. And that portion, the blinded step 1 and step 2, continued for a total of 3 years after randomization. If people were randomized to group B, the Truvada arm on the bottom, they, for the first 5 weeks, took a placebo cabotegravir tablet and an active Truvada tablet. And then that active Truvada tablet continued And when they got to the injection phase of the study, again, if they had acceptable tolerability in the first part, they were receiving placebo injections on the same schedule that, that people were receiving the active cabotegravir injections in the group A orange arm. If somebody stopped their injections early or they got to the 3-year time point, everyone was offered a year of open label Truvada after they completed their last injection. Next slide, please. These are the sites at which HPTN 083 enrolled participants. Ultimately, the study enrolled 4,570 participants at 43 sites. There were 27 sites in the United States, 5 sites in Peru, 4 sites in Brazil, 2 sites in Argentina, 1 site in South Africa, 3 sites in Thailand, and 1 site in Vietnam that conducted the study. Next slide. So this is what Dr. Grinstein and I were referring to, that we are so proud of and so delighted with the incredible partnership with community in the design and enrollment and conduct of this study. This is, as far as I am aware, the efficacy study of a prevention product in HIV that has had the greatest representation of key populations and most vulnerable and at-risk populations to make these results generalizable to the populations who need additional HIV prevention options the most. The analysis population included 4,565 cisgender men who have sex with men and transgender women who have sex with men, whose average age was 28 years. Fully 2/3 of the study population were under the age of 30. We had pre-specified that at least 50% would be under the age of 30, so we exceeded that. 40% met the definition of youth or young adults by most research definitions, that being under the age of 25. That wasn't pre-specified, but we're really, really happy and proud that we were able to engage with young people and adolescents globally. The protocol had specified that at least 10% of our enrollment would be transgender women. We were able to engage with and enroll 12% of the overall population, trans women. Of the US enrollment, we had pre-specified that 50% of the enrollment or more would self-identify as African American or Black, and we were able to meet that metric. Next slide. So the primary study questions that the study was trying to answer were: how effective is cabotegravir when compared to Truvada, and how safe is cabotegravir when compared to Truvada? Of course, we're only able to make comparisons between the 2 drugs in this study because there is no placebo arm by intent, so we cannot compare it to a placebo. We can make some speculations about a hypothetical placebo, but we're not able in this trial to compare this to a placebo because such a trial would not be ethical with effective HIV prevention products. Next slide. Okay, now we're going to get to the preliminary results and a little bit of context first. So I think most are aware that randomized blinded trials like this always are reviewed by a data and safety monitoring board called a DSMB. This DSMB resides at the NIH. It's a multinational group of experts that is independent from the study conduct, that advises the HPTN 083 protocol team. Their responsibilities are listed here. They're responsible for periodic review and evaluation of unblinded study data for safety, conduct, progress, and efficacy. Importantly, this group has met every 6 months since the beginning of the 083 study, reviewing unblinded data, which means for all of these parameters, safety, efficacy, et cetera, they are the only ones who see these parameters by arm. So they see how many HIV infections were occurring in the Truvada arm and how many were occurring in the cabotegravir arm. Similarly, all of our safety data, they are able to see by arm, by Truvada versus cabotegravir arm, whereas none of the rest of the people in the trial have access to that information. So we are really reliant on the DSMB to inform us if there is an imbalance either in safety and efficacy between the arms that is either clinically or statistically significant. They then make recommendations to the Division of AIDS at NIH as the sponsor regarding whether the study should continue, should be modified, or should be stopped. Next slide. So these are the efficacy results of the study. And I just have to say, I adore this slide because I think the image on this slide really speaks to empowerment of community to take charge and ownership of their own protection against HIV, and it highlights the youthful population that we were targeting and successfully enrolled into this study. And I love this image. So I particularly want to thank the community program managers and communications group at HPTN for creating this image for us. These are the preliminary results. The HIV incidence rate in the study was extremely low. It was 0.79%. And what I interpret this to be is that this indicates that both products in this study were highly effective for HIV prevention in the HPTN 083 population. We do not have a known background incidence rate for this population because, as I mentioned, there was no placebo arm. We were intending to enroll a population with an estimated incidence rate of somewhere around 4.5%. We do not know if that is, in fact, the background incidence rate in the population that we did enroll, but if you assume that it was somewhere in that range, that is an impressive and dramatic reduction in HIV incidence. We, of course, would like it to be as low as possible. We would like it to be 0, but 0.79% is a low number in comparison. So we were very, very excited by that information. What's really important in interpreting these results is that there are certain things we know about the comparator arm, the Truvada arm. We know that Truvada is highly protective against HIV acquisition when taken as prescribed, and it also was in this study. The incidence rate among the individuals in the study who were randomized to the Truvada arm was 1.21%. So again, a very low number, as would be expected in an at-risk population who was taking a highly potent HIV prevention product. As we talk about the results, I think it's really important that we remember that Truvada is known to be highly protective and that any comparison made to it had an incredibly high bar to be compared against. That is the known efficacy of Truvada in this population. Even with that in mind, in an absolute count number, there were 3 times as many HIV infections in the participants who were randomized and received the daily oral Truvada as in those who received the cabotegravir arm that included the injectable product. The incidence rate in the cabotegravir arm was 0.38%. There were 12 people in the cabotegravir arm who acquired new or incident HIV infection compared to 38 individuals in the Truvada arm who were found to have new or incident HIV infections. From a statistical standpoint, this result is defined as non-inferior. We were not able to demonstrate statistical superiority in this study based on these preliminary results. Please remember, the final results of this interim analysis are still pending. So we cannot determine statistically that the cabotegravir was superior to Truvada. However, the absolute numbers are, as I mentioned moments ago. Next slide, please. What's really important to know in understanding these results is that the participants who were randomized to the Truvada arm were adherent to the daily oral Truvada. What we know from the initial look at the data is that 87% of a small adherence sample, about 400 people that we've looked at so far, had detectable tenofovir in their plasma samples. 75% of those samples had levels in the plasma consistent with daily dosing of Truvada. What we have not been able to look at yet, it's in progress, is the adherence patterns of the individuals who were randomized to the Truvada arm who did acquire HIV. So those obviously are an incredible priority for us to look at those and fully understand what the adherence patterns were for the people who did acquire HIV in the Truvada arm, those 38 individuals. Similarly, we are measuring the levels in the blood of cabotegravir in all of the participants who acquired HIV when randomized to the cabotegravir arm. We're also looking very carefully at the timing of those infections compared to what we know about the last dosing of cabotegravir that those participants received, since that's administered in the clinic, and we will be able to identify the dates of last administration of cabotegravir injections very precisely. We do not have that information at this time. As soon as we have that information, we look forward to sharing it with you all. Next slide. We are, of course, going to be doing resistance testing on all of the people who acquired HIV in this trial, since with the prolonged pharmacokinetic tail that we know is associated with cabotegravir, that is a concern for communities, for providers, for policymakers, for regulators, and for the manufacturers of cabotegravir. So we will be looking very carefully at the resistance in everyone who acquired HIV on the study, regardless of which arm they're assigned to. Again, I do not have that information available today, but we look forward to sharing it with you as soon as it is available. Next slide. In terms of safety, we were delighted to see that, again, in this preliminary data cut that we have at this time, that injectable cabotegravir and the daily oral Truvada used as PrEP were similarly safe and well tolerated. There were 3 adverse events, 3 side effects that appeared to be more common in people who received the cabotegravir-based regimen, and those were injection site reactions. 80% of people who received cabotegravir injections have reported at least some injection site reaction of any severity, compared to 30% of the people who were receiving the placebo injection and the active Truvada reporting any injection site associated side effects. There were also more individuals in the cabotegravir arm who reported a raised body temperature, or called pyrexia, or a mild fever, and also high blood pressure, not severe, mild or moderate elevations in blood pressure, but those events were more common in the cabotegravir arm compared to the Truvada arm. What I do not know today is if those elevated body temperature reports and elevated blood pressure reports were in people who had pain or discomfort from their injections. That would make sense intuitively, but we're looking into that, and I hope to be able to report back on that to you in the future when we have the final analysis available. Yes, so just before we move to the next steps, I do want to make one more comment about the injection site reactions, because although I'm seeing a number of comments in the chat box that people are surprised that 80% of people randomized to the cabotegravir injections had any level of injection site side effects. That is consistent with the cabotegravir when used with long-acting injectable rilpivirine for HIV treatment or maintenance of virologic suppression. The majority of people report some pain or discomfort at the site. The good news is it's overwhelmingly mild or moderate, and very few people had to discontinue the injections because the discomfort was so significant. In our study, only 46 participants out of all the people receiving the injections, so just around 2% of all the people receiving any active cabotegravir injections were the discomfort from the injections so significant that they didn't want to take any more injections. So that's, I hope, a little bit of context. So just so everyone knows, what we are doing as a study team right now is we are informing all of the study participants. That's in process. We have letters to participants and an amendment pending at all IRBs and ethics committees that are reviewing the study at sites. They're in the process of being reviewed and approved there, so that information can be pushed out to participants as quickly as possible. Site investigators have been made aware of these results already, so they can have conversations with participants and get this information out. We are unblinding all our participants. Again, that unblinding activity has to wait for local IRBs and ethics committees to approve that unblinding, but the information is available at the sites as soon as those approvals go through at each site. Participants will be allowed to stop their placebo medications. They will continue on the active product that they have been taking in the study at this time. And as soon as we are able to get sufficient cabotegravir to the sites, we are going to offer that to all participants in the study. And we hope to understand the timeline of when that will be able to be implemented in the next couple of days to weeks. As soon as we know the timeline, we will make sure that we are transparent about letting people know how quickly we think we're going to be able to make that available to people. People who don't want cabotegravir and just want to stay on their Truvada in the study will be allowed to do so until the original 3 years since randomization is up, and they will be able to stay on that. The duration of the follow-up that people will be able to access CAB-LA in the protocol is not yet determined. And again, I'm not trying to be untransparent. There are just a lot of conversations going on right now about the duration of that open label extension, but everyone is committed to making open label cabotegravir available to participants in 083 who want it for at least some amount of time after this. Again, full duration of that availability to be determined. And participants for the moment will be continued to be followed on the current 083 protocol. Again, we will be amending it to be an open label extension. Okay, next slide. So those are the preliminary results. I just want to take a moment at the end of this presentation to really thank some studies take a village, some take a township. This really took a metropolis to conduct. And so I really just want to take a minute to give our heartfelt thanks to the sponsors, the Division of AIDS at DNIH, NIAID, NIMH, NIDA, with additional funding from ViiV, all of the critical components at the HIV Prevention Trials Network, including the Laboratory Center, the SDMC, the Leadership and Operations Center at FHI 360, and HPTN leadership, who have been so unequivocally supportive of us in conducting this protocol. Pharmaceutical support from ViiV and Gilead, our colleagues at AIDS, our HPTN leadership listed there. Next slide, please. The folks at the Leadership and Operations Center, especially Mary Beth McCauley, Kyla and Andrea at the LOC, our incredible community team, including Cheryl, Jonathan, Marcus, who led us in this community engagement process, our communications team, our finance team, the folks at the Lab Center and the SDMC, our team members who work tirelessly, our community educators, our recruiters, and our CAB members. Next slide. And finally, our sites, our incredible site teams and investigators, and most of all, the incredible participants who trusted us with this study, believed that the results would be relevant to them and their communities, took a chance on a complicated, long, and involved study, and really are the winners in this study, as we hope that this will lead to regulatory approvals and widespread equitable scale-up of an additional prevention tool, should the review of this study support so on rigorous peer review and regulatory review. So thank you. This is a shared win for all of us, and I hope everyone on this call can feel pride and excitement for data that we hope will stand up to peer review and regulatory oversight, and will lead to enormous steps towards dramatically reducing HIV infections, if not ending the epidemic outright. Of course, to end the epidemic outright, we will need the critical results from the 084 study that we are now going to hear about. And I'll stop there, and thank you very much for your attention.
Great. Rafi and Beatrice, thank you both so much. Congratulations to all of the people, and particularly the participants on the slides as you described. Rafi, when you do win a Tony Award, as I know you will one day, I can't wait to hear that speech as well. But really, that was beautifully said. And your last point was the perfect segue to introduce Sinead Delaney-Moekle and Mira Hassanpour, who are the co-PIs for the sibling study, HPTN 084. Sinead, based in South Africa, and Mina, based in Malawi, to talk about that critical study. There are enormous number of questions that are already here. We're going to get to as many as we can. I want to encourage people to keep sending them as we go. I'm trying to orient them. But Sinead and Mina, over to you.
Thanks very much, Mitchell, and thanks very much for giving us the opportunity to talk to you about 084. I think we want to start off just by congratulating the 083 team and the participants in 083, and saying that it provides huge inspiration to the 084 team to answer the similar question in women. Next slide. So the DSMB met to review the HPTN 084 study on the 14th of May, and they had recommendations for 084. Next slide. 084 is a very similar study to 083. As you will have heard Dr. Landowitz explain, participants in 084 are randomized either to receive active cabotegravir and matching placebo for Truvada, or active Truvada and matching placebo cabotegravir. And they move through 3 steps: a 5-week oral lead-in phase, followed by an injection step where they receive injections and blinded study pills, the pills which they take daily and the injections which they receive 8 weekly after an initial 4-week run-in. And then for participants who discontinue injections, they are offered 48 weeks of oral Truvada. Next slide. So when the DSMB reviewed 084, we had enrolled 3,032 participants out of a target 3,200, which was 95% of our enrollment target. At that point, we had been paused to enrollment in response to the COVID pandemic. But participants had been recruited at 20 sites in 7 countries in Eastern and Southern Africa, and those countries are Uganda, Kenya, Malawi, Zimbabwe, Eswatini, South Africa, and Botswana. And to date, amongst the participants enrolled, they had a median of 25 years of age, and half of them obviously were under the age of 25. And all participants would have been PrEP eligible using a score that predicts HIV incidence in the absence of PrEP. And unlike 083, because this trial enrolls cisgender women, all participants were required to use a long-acting reversible contraceptive, that is an injectable, an implant, or an IUCD, to prevent pregnancy. Next slide. So how do 084 and 083 differ? Well, they're 2 very similar protocols, and you've heard that they are sibling studies. But one of the key differences between 084 and 083 is that 084 aims to answer the question about whether cabotegravir compared to Truvada is more effective whether CAB is more effective than Truvada in preventing HIV infection. And part of the reason that we ask this question is because given the previous results of Truvada, the placebo-controlled trial results where Truvada was evaluated in women, the range of estimates of effectiveness were highly variable, which meant that we couldn't take the same path as 083, and we've had to frame our question in this particular way. Next slide. So when the DSMB reviewed 084 this month, we had accrued 25% of our person years. So this is half of what 083 had accrued. And this is because we started 084 a little bit later than 083. But also, if you recall, we had a period of enrollment pause in order to allow us to respond to the neural tube defect issue, which arose around questions related to dolutegravir. And so for this reason, we have accrued fewer data at this review. Importantly, the DSMB confirmed that they had no safety concerns about the study product in cisgender women in 084. Both cabotegravir and Truvada are deemed to be safe based on the data that they've reviewed, and they will continue to review safety at ongoing reviews. And so the DSMB recommended that we continue the study as planned. Next slide. So in terms of the next steps, the HPTN 084 study will continue to collect safety and efficacy data in cisgender women. And this is particularly because we are not sure whether the data observed in cisgender men and trans women will translate to cisgender women. It's possible that given the different routes of HIV infection, but also given the different metabolism of these drugs in men and women, that there may be differences. And it's important that we provide robust data in order to ensure licensure for this product for cisgender women. So in terms of the practical next steps, we will share these results with 084 study participants, as well as the communities from which they have been recruited, and with the local regulatory authorities that are overseeing the conduct of these studies in the 7 countries in sub-Saharan Africa. Next slide. So in addition, there are some additional ongoing studies which are critically important. There are 2 adolescent bridging studies, HPTN 083-01 and 084-01, which are paused to begin enrollment and will provide critical safety data in adolescent and young adult populations to support the regulatory approvals in younger populations. And in addition, there is a qualitative sub-study of 083, which will continue to explore barriers to sustained and consistent use of long-acting cabotegravir. And Mitchell, I think I'm going to hand back to you at this point, but just to highlight that there are additional resources which are available, which provide information both about 083 and 084. And then also just to acknowledge the sponsors and the donors and the support that we have received for 084 from the Division of AIDS, as well as Vive Healthcare and the Bill and Melinda Gates Foundation, and to acknowledge the ongoing contributions of our colleagues at in the HIV Prevention Trials Network, as well as the spectacular work of the sites in Eastern and Southern Africa, who have demonstrated huge commitments to continuing the study despite the COVID pandemic, and who are truly committed to answering the question about whether cabotegravir is safe and effective in preventing HIV infection in cisgender women. So we really appreciate the contributions of the sites, our community educators, and the CAB members, and the communities from which participants are drawn. Thank you very much.
Great. Thank you so much, Sinead, and on behalf of Mina and the whole 084 team. Really, really grateful. There are so many questions, and we have about, well, I'm going to cheat and say 15 minutes, and maybe we'll go a little bit over if that's okay with everybody. Get to as many as we can, and I know there's a commitment from the HPTN and AVAC can help to make sure that these questions get cataloged and answered. As Sinead said, there are a number of materials that are already being posted, as will a recording of this call. And a quick question, I guess, for Sinead and Rafi, these slides will be available as well? People were asking.
Yes.
That's
Great.
correct.
Beautiful. I wonder, maybe I'll just start because it came up very early, and Rafi, you actually already answered it, but I think there was a fair bit of confusion this week about what the actual accurate way to describe the results, and you did it, I thought, so well. But I just want to make sure that you're able to say again how you're describing the result, particularly in terms of the comparison to oral PrEP, and this issue that, you know, statistically non-inferiority, superiority, and a question came up, although stopped now because of non-inferiority, could you possibly see superiority when the data are fully analyzed? And yet, how would you describe the results now?
Right. So that's a great question, and I know everyone on this call thinks a lot about these sorts of issues very carefully. So I'm going to sort of tell you how I think about it. What the DSMB said to us when they recommended stopping the study last Thursday, because that's when all this happened, was they said, you have clearly shown statistical non-inferiority, and you have approached superiority. You did not show superiority, you approached it. You were very close. And so to me, that is an exciting result, but it does not allow us to definitively say, based on these interim results, that one product is better than the other. That's what superiority would let you say. Non-inferiority just tells you that the products performed similarly, and technically in the statistical language, what it means is it's not worse than the comparator product by a certain amount. In this case, the margin that was used for this non-inferiority was 23%. So what one can say with confidence, with extreme confidence, it's definitely not more than 23% worse than Truvada. The way I look at it is they both performed very well. Numerically, the point estimate was that there were fewer people who acquired HIV when offered cabotegravir than in our study in the arm of people who were offered Truvada. From a regulatory standpoint, that's not inferiority, it's not superiority, and that's what we have to say. That's the correct interpretation of it based on what could have happened just by chance. We are working on the final analysis. It's statistically fraught and complicated and nuanced whether or not you can ultimately claim superiority on the analysis of the final information. If we feel, after a lot of discussion and looking carefully at the data, we can confidently state that we have a superiority result, we will do so, and we will present that in a peer-reviewed context and make everyone aware of that. But at this time, the study was stopped for non-inferiority, approaching superiority, which is exciting, but doesn't meet that extremely rigorous metric. I would also just remind people that the study was originally powered to require almost 4 times as many HIV infection events to be fully powered. The fact that it was stopped this early at this time point when only a quarter of the expected HIV infections had been observed really indicates the strength of the evidence, the potent evidence of great activity by both products in this study.
Great. Thank you, Rafi. So clear, and I know it has been a source of confusion, and really important that we continue to remind people of that. A bunch of questions, obviously, about what's next, and you did address a number of those things, but some questions about both what will happen with participants in 083 in terms of being able to choose, and you mentioned that they'll stop the placebo part of their regimen, and that you mentioned that those on oral PrEP you would offer them cabotegravir as the injectable, but questioning about whether people can go the other way, even though it was shown to be safe and effective. If someone doesn't want to be on an injection every 2 months, can they switch to oral PrEP? And another related question, what is the kind of social behavioral science work that you could do to really look at some of those determinants of product selection now in a very real world way?
Yeah, thanks for those great questions. So yes, as part of the 083 protocol, and that will stay, right, as an injection washes out of the system, you would want to make sure someone who's ongoingly at risk has a protective active PrEP agent in their system. Whether or not we'll be able to offer people the oral pill form of cabotegravir as an alternative for covering that table or not is something that we're discussing. If not, then the protocol does permit people to go on open label Truvada for a year after they stop those injections. So that part of the protocol is not changing, that there will be some form of opportunity for an oral based PrEP after a last injection to come off it in a safe way. We do have qualitative sub-study that's ongoing, and we're going to be reconvening to think what qualitative and quantitative evaluations of this opportunity for choice we can take advantage of and learn from this open label process now that we've demonstrated potent efficacy for both products. And I really just want to emphasize that people who are on Truvada and want to stay on Truvada will be able to do so until that 3-year time point. And I think I mentioned that previously.
Great. Thank you so much. Incredibly helpful. There are always have been questions in these studies about both the oral run-in and the tail that you just mentioned. When someone stops with the injections, they're in the protocol, what you described as that step 3. Do you have, I realize it's only a week into this since the DSMB, but any insights yet about the role of both an oral run-in and a tail cover as we look to actual potential implementation should this product get approved? And if not, what might be the pathway of additional data analysis coming from both studies to help address that?
Yeah, thanks. Incredibly important questions. I think we all think conceptually that the power of a long-acting product is you don't have to take a daily pill. And so the concept of the oral lead-in was something that I think frustrated a lot of people who are interested in the long-acting injectable. I know both regulators and ViiV and all of us are interested in understanding if it is safe to go, quote, direct to inject. And I believe there are going to be some data in the HIV treatment field about one of the ViiV-sponsored treatment studies that did have a cohort of patients who didn't go to an oral lead-in before getting the cabotegravir rilpivirine injections. And I don't know those data yet, but if that is confirmed to have been safe to do, I think there's going to be increasing confidence that it might be safe to do in the PrEP context also. We're obviously going to be looking really carefully at the rate of side effects and safety events that happened in that step 1 oral lead-in in 083 to better inform if that seems like it's going to be a required element. We would love it if it were not a required element, that it could be made optional or removed entirely. But again, the data, we haven't had a chance to dig deeply enough into it yet to know the answer to that. And then flipping to the other part of your question, you asked about the tail. I think the tail and understanding its clinical relevance, what happens if somebody is exposed to HIV and acquires HIV during various points in the tail, what that means for resistance, what that means for drug-drug interactions, what that means for, in cisgender women's setting, for fertility, for potential for pregnancy, breastfeeding. All of those issues are critically important. And I think the 083 and 084 tail phase data are going to be just a drop in the bucket of the kind of information we're going to need as this moves forward. I think they're going to need to be registries of tail phase experience as this moves forward. And we're going to need to think carefully about how to have centralized ways to monitor and survey that, because I fear that even with 083 and 084 and the open label extension that will follow 083 and we hope 084, if it's similarly successful, or I should say when it's similarly successful, won't be enough of an experience to really be conclusive as to the significance of the tail. So I think it's a story that we need to continue to be vigilant and careful about and look towards.
Great. Incredibly helpful. I realize we're just getting to the bottom of the hour, but I still see lots of people on, and assuming that the trial leaders are on in the PTNs, okay, I'm going to keep going unless I hear violent objection, at least for a couple of minutes before I turn it over to have the call closed out. I do want to get to one quick issue and then maybe some longer term issues. There was a quick question. Obviously, a lot of people on the call follow oral PrEP very closely. One of the big things that began to emerge shortly after IPREX was trying to understand the quote unquote time to protection. And there we eventually found out that about 7 days. Do you have a sense yet at all if there is some correlation? Is that first injection you're good to go, or do you have any sense of either, do you already know time to protection from first injection, or will that be something that still can come out of the full analysis of 083?
Yeah, it's a great question. It's a nuanced question. It's a critical question. And it's something that we're going to be interrogating the data as best we can to see if with this small number of infections, we are able to get sufficient information to answer it from these clinical data, or if they're enough to support a modeling exercise that could help us answer that. But obviously, understanding the timing of onset of protection, the waning of protection after last use, and all of the adherence-related kind of understanding that we now have with great granularity with Truvada, we need to reinvestigate with cabotegravir, and we just don't know that yet. But it's that information, the resistance information, and the timing of these infections relative to previous adherence are the top priorities that we have for the immediate analysis of this data set.
Great. I do want to make sure there were lots of notes in the chat. I'm sure that the PTN will save it. But enormous congratulations and kudos to the team for the diversity of enrollment and also to have both of these studies designed up front to get us a product for all populations at risk. But a question came up about, is it truly all populations at risk? And questions about what we won't know from either of these studies related to trans men, to heterosexual men at risk, and people who inject drugs. And just wonder if any of the four of you, knowing that those are not populations in the current trials, what that means for the product writ large, and maybe particularly Sinead, thinking about the African context, absolutely needing options for women at risk, but also heterosexual men at risk. And just wonder if any of you want to comment on that.
Yeah. Sinead, do you want to go first?
Thanks, Mitchell. I mean, I think that's an incredibly important question. Certainly, there has been a first attempt to try to answer the questions about the safety and efficacy of CAB in at-risk populations, with the idea that representing a diversity of populations can ensure a broad licensing label. I think we've seen the importance of to the regulators of returning as much data as possible, which is why you know, kind of there has been an attempt to ensure that we reflect these populations as well as include data on adolescents. And I think the kind of the potential exclusion of other populations is not a deliberate attempt to exclude, but rather a strategy to answer these questions in populations that are at highest risk so that we can very rapidly get these products, demonstrate their safety, efficacy, and potentially proceed to licensure. So that is my reflection on this. But Rafi, I'm sure you have additional comments.
Well, the only additional comments I would make is I think this is now an opportunity to expand the scope of at-risk populations to be investigated. I do think we can make some generalization to heterosexual men when we have the breakdown of the 083 population who reported only engaging in insertive intercourse. And I think that will be at least suggestive of the essentially penile protection of this product. I think we definitely need data in those other populations, as Sinead noted.
You know, really, really helpful. And I'm reminded from the oral prep experience and something that Jared Bateman and Connie Kellum said after Partners Prep, and there's populations in clinical trials, and then there's what we're trying to do with prevention products and programs writ large, and it can take a suite of studies to get us everything we need, and just want to, history may not repeat itself, but it rhymes a lot. There are an enormous number of questions that won't surprise anyone that come into the larger what next, beyond 084, about costs, both for the product and its delivery, related to the licensure plan. Obviously, this is not the HPTN's product, and I'm not sure who, if anyone from ViiV is on the line, but perhaps a chance, given that we have such a large number of people here, I think it is important for people to understand maybe some of that process that unfolds. I don't know, Rafi, Beatrice, Mina, and Sinead, if any of you feel comfortable commenting on that, and at least to the degree that you can as investigators of these pivotal studies.
Well, we've been deferring all of those questions about those issues to ViiV, who would be the one deciding on the timing of regulatory submissions. And we don't know at this point if the plan is to wait for the 084 data or engage in discussions around a single population approval with a second one to follow. So I don't know if anyone from ViiV has joined the call, but I would defer any comments with any substantiated information to them.
Great, thanks, Rafi. I'll pause just for a minute to see if anybody does want to unmute. I know we're hoping to organize a follow-up separate call about the pathways forward, which we can talk about in a minute. But I'll pause for a minute for anyone from ViiV wanting to jump in. All right, I will say that, there is, we obviously have the experience very recently with the FDA approval of TAF for PrEP. Done based on one trial in one or a couple of populations, but no other trial was ongoing. This is a quite a different situation where these trials were designed as a combination suite up front. So we will look forward to that. I am confident we will want to understand that all as a global community of advocates and researchers with policymakers and with ViiV to understand the pathways forward. And recognizing too, we're a week out from an unexpected early result. So going to be really important to flag that going forward. There were a bunch of questions too.
Mitchell,
oh, sorry, go ahead.
Kimberly Smith from ViiV is on, but she says she's not able to speak. Just wanted to point that out.
Okay, no problem. It is, and I, obviously these are huge question marks for everybody. We will continue to find ways to have those conversations as more is known. And thanks, Meredith. There were a bunch of questions about demonstration projects beyond the current trials and the open label extensions and some of the additional studies that Sinead described. I will just say very quickly that there is an effort that a number of people on the line have been involved with through a Gates Foundation funded initiative under a project we lead at AVAC called the HIV Prevention Market Manager, that is looking at biomedical prevention introduction collaborative, the so-called BioPIC, that's been working for the last 2 years with a number of key stakeholders to think about a introduction strategy that would include demonstration projects and full-scale introduction when the product is shown to be safe and effective in all populations, hopefully. That work actually now obviously needs to be accelerated. The whole driver there is to be sure that we don't have the delays that we've had with oral PrEP, shown now almost 10 years ago in IPREX to be safe and effective, 8 years since licensure. And we all hope we can be a little more strategic and faster off the mark once licensure is provided.
Hi, Mitchell. This is Kimberly Smith. I'm unmuted now, I think.
Go ahead.
Hi, how are you?
Very well.
So this is Kimberly Smith. I'm head of research and development for ViiV, and I just wanted to answer the question that someone was asking with regard to plans for regulatory submissions. And so, as we've stated previously, it is our intention to file as soon as possible, ideally with men and women, because that is our goal, to get approval for men and women. So we are very much anticipating the results of 084, and we are in conversations with the FDA as well as other regulatory agencies around the world to make a plan for submission. So hopefully that answers the question.
That's super helpful. I appreciate that. We are now a fair bit over time, and I want to just commit that these amazing questions and comments will continue to be cataloged, and we'll be looking for how to get back to everybody with responses, some that can be answered, some that can't. One quick question, Rafi and Beatrice. Obviously, there's still a lot of analysis to be done. Do you have a timeline in terms of when you think we might see publications or presentations of the data? I don't know if you mentioned that.
No, I didn't, but we're obviously hopeful that we will be presenting at a major HIV conference in the not too distant future. And we're working on a manuscript immediately. I can't give you a time frame on that, but it's proceeding as fast as we can carry it along. So we look forward to getting this out for peer review so that it can be held up to scrutiny and then hopefully moved out into the public domain with appropriate levels of scrutiny and review.
Fantastic. Maybe you got the email, Rafi. I think late breakers are still open for AIDS 2020. Maybe
oh my God, Mitchell, are they?
Fascinating. And good timing perhaps, but we will all look forward to that. I am in just a minute gonna turn this over to Melissa Turner, who is the chair of the community working group of the HPTN. And as you heard from Beatrice at the outset, community engagement and through both the working group in the PTN and working with civil society partners throughout is critical to all prevention research. It's as important as the products we test and the sites that run trials and the laboratories that look at samples. And before I turn it over to her, I do want to just see Mina, Sinead, Beatrice, or Rafi, any last comments from any of you before I turn it over to Melissa.
I guess the only thing I would say is thank you again to you, Mitchell, and to Melissa and the PTN community program managers for organizing this opportunity to share this and introduce these data to the community, engage in this important conversation. And we look forward to sharing more and having more conversations.
Great. Others on the trial teams?
I think just to echo what Rafi has said, and on behalf of the 084 team, thanks very much to you, Mitchell, and to Melissa, and to the HPTN team and others who have put this together. It's really helpful to be able to share the 084 recommendation and to hear the support to continue this study in women in order to deliver a similar robust result.
Great. Thank you. And also, I say there were a bunch of questions too that I totally forgot because we really didn't have time. Obviously, one of the other things in addition to 083 and 084 and the potential of cabotegravir is, of course, the implications of these results on the next generation of trials, because no matter how good any of our interventions are, probably the most important word throughout all of these conversations is options. And we need them all, and how these results might influence next generation trial design. So there are obviously a series of calls that we all need to have. And also I did want to just put on the record for recording an incredibly important recommendation from Nita Marar in South Africa about setting up also a call for, given the importance, as the team described, of community, the importance of a call with community advisory board representatives and maybe even participants themselves to describe their experiences. I think that was a great suggestion, Nita. So there are clearly a lot of calls to have about future access, the trial experience from the user perspective, and implications for next generation research. And I'm sure there are many others, and I know we certainly look forward to working with the HPTN on all of that. But before we finally close, I do want to turn it over to Melissa from the Community Working Group of HPTN and to give her the last word.
Good day, good day. I hope I'm being heard. Am I being heard?
You are, loudly and clearly.
Excellent, excellent. I've been in some kind of audio Zoom purgatory. I do apologize. I want to thank everyone on this call. I just want to take a moment to reiterate the gratitude that has been expressed on today's call. This is a celebratory moment, exhilarating moment, really, to discover evidence of a new tool. But at the same time, it's deeply, we feel deeply humble and gratitude as a network, and we just want to extend again our recognition to all of the community partners, the collaborators, the stakeholders, and certainly those participants, thousands of participants who have trusted the clinical research site staff teams and engaged in these studies and given of themselves to advance prevention science. We want to be sure to just lift them up and give them thanks. And as a network, we continue to be eager to move forward. We continue to be hungry to press forward, to dig for the next scientific finding, the next breakthrough. We are not going to rest on any of our attainments to date. We want to press forward, and we want to work continually with our partners towards those ends. And I hear the recommendation that we need to have additional calls to engage the community, the participants, the community advisory board members. We will be sure to do that as is consistent with the guiding principles of community engagement, respect, transparency, that mutuality and understanding. We want to be fully communicative and transparent with our stakeholders and all of those who are invested in this science. And so we will move forward with those goals. And we just are grateful to all of you for partnering with us, the HPTN network, as we are striving to reduce incidence worldwide of HIV. Thank you all. I appreciate your patience. Thank you. And particularly thank you to you today for carrying forward with the facilitation of this call, Mitchell Warren. We are so grateful to you and AVAC for your treasured collaboration with us. Thank you. So that will close today's calls, and I know we've gone over time, and again, we'll have future updates, and thank you one and all, and have a beautiful day.
Thanks all. Stay safe and well.